Hey, hello everyone. Today, Jennifer is going to talk about GenStore, which was published in ASPLOS 2022. She's second semester in computer science and has experience in biology by having bachelor's and master's degree at ETH. So she is combining her knowledge and also presenting this paper. So it's a very good fit. So good luck, Jennifer. And yeah, the stage is yours.
Right, thank you, Nika. Okay, hi everyone. Today I'm presenting a review of the paper GenStore, a high-performance in-storage processing system for genome sequence analysis. At first glance, we can already see from the title that this paper relates to several topics we have covered in this class. It enhances genome sequence analysis, for which we heard an introduction by Mohammed a few weeks ago, and it implements processing in storage, which relates to the issue of high data movement cost. The paper was published by researchers in the Safari group very recently, that is a few weeks ago at the Multidisciplinary Computer Science Conference ASPLOS, actually at the same one as Gianluca's paper before. In one sentence, this work reduces movement of genomic data from storage to main memory and CPU by performing part of the computations directly in storage and filtering which data need to leave storage for more expensive processing. A few words on why I chose this paper. Before I studied computer science, I studied and worked in biology, so I already know genome analysis from a biologist's perspective. And choosing this paper gave me insight into challenges with genome sequencing from a computer scientist's perspective. Because at the time I left the field of biology, it was already the case that not data generation in the lab, but data processing was the limiting step in genome analysis. In reading this paper, I found out why this is the case and what can be done to accelerate processing of genomic data. So let's look at an overview of the paper in form of the executive summary. The COVID pandemic is just one example of the growing importance of genome sequence analysis. In the processing of genomic data, read mapping is a fundamental step that often bottlenecks processing because it's computationally expensive and performed on a large amount of data. There have been significant efforts to improve read mapping, but while these approaches reduce the amount of expensive computation, movement of the data from storage to the processing unit still imposes a large data movement overhead. The goal of this paper is to improve genome sequence analysis by reducing unnecessary data movement from storage. And the key idea is to filter out data in storage that does not require expensive read mapping and thereby reduce the amount of data movement. Challenges that the authors met were different properties of the read mapping workloads and limited hardware resources in the storage system. In the paper, they presented GenStore, the first in-storage processing system that reduces both computation and data movement overhead in read mapping. And evaluation shows that GenStore significantly speeds up read mapping and reduces energy consumption. So this brings me to the outline of my talk. I will start by giving a bit of background information on read mapping and then summarize the goal, key ideas, and key results of the paper. And since this is a paper review, I then move on to the analysis, where I will point out a few strengths and weaknesses of the paper, and finally end by listing some takeaways. At the end, we will hopefully still have some time to discuss your questions and a few other topics I prepared. Let's start by remembering the workflow for genome sequencing. We start by collecting the data sample and preparing it in the lab for sequencing. Either before or during sequencing in the sequencing machine, the DNA will be fragmented. Where originally we had a few very long chromosomes, we now have relatively short stretches of DNA, for which we no longer know where they come from in the genome. Now we have the sequences of short DNA stretches called reads and have to find out which location they had in the original genome. This is done by the process called read mapping, where we use a reference genome to map each read to potential locations in the reference genome. And this step is computationally very expensive because it has to account for differences between reads and reference genome in terms of alignment scores. Also, it has to be done on a huge amount of reads. Finally, depending on the biological question, the data is processed further. I included this figure from Mohammed's talk to emphasize that read mapping is a central step in processing of genomic data in virtually all types of biological questions. I said already in the beginning that genome sequence analysis is limited not by data generation, but by data processing, which is due to huge performance increases in sequencing technology. If we look even closer, which step is the bottleneck in data processing, we see that read mapping makes up a large part of the analysis time. For example, in a study to discover sequence variants in cancer genomics, read mapping accounted for 45% of execution time. So what's happening during the step that it takes so long? Conceptually, the goal of read mapping is to find matching locations in the reference genome for each read. This takes long because one, this requires an expensive dynamic programming algorithm to determine the alignment score, and two, it has to be done for a huge number of reads and locations. As a reference, the human genome has 3 billion base pairs, which corresponds to hundreds of millions of reads. Let me go into a bit more detail about the read mapping process. To make the comparison of the reads and the reference genome easier by reducing the search space, the reference genome is stored as an index structure, where all unique sequences of length k are stored together with their locations in the genome. That way, it's convenient to look up sequences read in a read in the index and get potential locations for them. Read mapping consists of 3 steps. The first step is called seeding, where the goal is to find potential matching locations in the reference genome. Here, the read mapper takes subsequences of length k from a read and looks them up in the index. Note that conventionally, k is shorter than the read length, as it's shown here, where k is 3, but the read length is 12. If there's a hit, the read mapper marks the potential locations in the genome. In the next step, called chaining, the read mapper reduces the number of potential locations by merging overlapping seeds into longer chains and removing locations with too few or too short chains. And if there are still potential locations for the read in the genome left, the third step starts, which is sequence alignment. Here, the read mapper determines the exact alignment score, that is edit distance between the read and each potential matching location using dynamic programming. And this step is computationally very expensive. Finally, the read mapper returns the location with the best alignment score for this read. And this process is repeated for each of the huge number of reads. So naturally, a lot of research effort has gone into improving read mapping, and previous works improved the sequence alignment process, designed hardware accelerators, or reduced the number of reads requiring the expensive sequence alignment step by filtering out unsuitable reads. However, so far, the genome data has always been transported from storage to main memory and the CPU to perform these improvements. And this has the disadvantage of having a large data movement overhead. So to study the potential of an in-storage filter solution, the authors did an experimental case study with different read mapping systems and different SSD storage configurations. In this case study, they made several observations. For one, if reads are filtered in storage, performance can be improved because data movement overhead is reduced. And also if filtering is performed out of storage, as in previous works, the filter competes for resources with sequence alignment. So based on these observations, what's the goal of this paper? The authors aimed at improving the performance of genome analysis by designing a filter that resides in storage and reduces the number of reads that have to go through the alignment process, and also reduces the data movement overhead from storage to the rest of the system. Let's also look at the design objectives they aimed for. Of course, the system should provide high performance in storage filtering, such that read mapping of unfiltered data in the rest of the system can be overlapped with filtering in storage. Second, as sequencing machines produce different read lengths and error rates, and organisms have varying genetic variation, the filter should be applicable to many different kinds of reads. And of course, the design should be cost effective. So the idea of filtering reads in storage sounds rather straightforward, but what are the challenges to achieve this? Let me start by giving you a brief overview of the organization of a modern SSD storage device. In the paper, they implemented their accelerator in NAND flash-based SSD, which is the most widely used type of SSD. A modern SSD consists of 3 components. First, in orange, the actual memory, which is organized in packages. Second, in green, the SSD controller, which communicates with the outside system and manages memory requests internally. One opportunity for an in-storage filter here is that the internal bandwidth, that means between the SSD controller and the memory packages over the channels, is higher than the external bandwidth, that means between the SSD controller and the host system. And third, in blue, there is a bit of internal DRAM in the SSD to store information about managing the requests. A challenge with implementing a filter in storage are the limited hardware resources. For example, limited internal DRAM capacity to store metadata needed for filtering, or that NAND flash-based SSD have poor performance in random memory accesses. Another challenge, which we look at next, are the different read mapping workloads arising from different sequencing technologies in genetic variations. Reads can be roughly classified into 2 categories, depending on which sequencing technique was used to produce them. One type of techniques produces short reads with very low error rate. Other techniques produce much longer reads that have a high error rate. The error rate during sequencing, of course, doesn't say anything about the genetic variation between the genome that's studied and the reference genome, but you can roughly say that the longer a read, the more likely it is that there are natural differences to the reference genome. Now, what does this mean for filtering of reads? There are 2 good filtering opportunities arising from the information above. First, exactly matching reads because they have perfect alignment to the reference genome. And which type of reads is suitable here? That would be short read sets with low genetic variation. And second, non-matching reads that will not align to any location in the reference genome and that do not qualify for sequence alignment. And candidates are long reads or short reads with high genetic variation. So now that we know the goal of the paper and the challenges in achieving it, let's look at the in-storage filter system called GenStore the authors came up with. They designed 2 in-storage filters, one called GenStore-EM for exactly matching reads, and one called GenStore-NM for non-matching reads. We start by looking at GenStore-EM. GenStore-EM filters reads that match exactly one or more sequences in the reference genome. That means their alignment score does not need to be determined by sequence alignment. As a reference, when sequencing human genomes, 80% of short reads match at least one location in the reference genome perfectly. However, for longer reads, due to genetic variation and many sequencing errors, it's very unlikely that they match any sequence in the reference genome exactly. That's why GenStore-EM is only applicable to short reads. And the hardware cost for this filter is low because only simple comparisons are needed. Now, the researchers faced a challenge when designing the filter. During the first step in read mapping, seeding, when you take a read and want to look up subsequences of length k in an index, you make several random accesses to the index per read because they depend on the random subsequences in the read. This is challenging because first, NAND flash-based SSD have low performance for random accesses. And second, the entire index has to be present in the internal DRAM of the SSD, but there's not enough space for that. To address the problem of the random memory accesses, the authors made 2 changes to the data structures. First, they set the length of k to be the length of the entire read, where before the k-mers were shorter than the read length, k is now the length of the read. That means the index now has read length keys. This way, instead of multiple lookups per read, now only one lookup is necessary per read. And second, they sorted the index and the reads alphabetically. Instead of random accesses to the index, now sequential accesses can be made, such that not all of the index has to be present in the internal DRAM. Instead, reads and index can be streamed into the DRAM batchwise. Here's an example of the new design of the data structures. On the left, you see the reads, and on the right, the index structure of the reference genome. Both are sorted alphabetically. So now, using an animation that I was able to borrow from my mentors, we can see on a small example how GenStore-EM filters exactly matching reads using the new data structures. It sequentially scans through reads and index table and compares the entries. If, as shown here, the current read and index kmer are identical, the read is recorded as exactly matching and filtered out. It does not leave storage. The filter moves on to the next read and kmer. If the read is alphabetically larger than the kmer, as G is alphabetically larger than C, there is no read in the read set that matches the kmer in the index, but the read might still match a later entry in the index. In this case, the filter just moves on to the next entry in the index. But if the kmer is alphabetically larger than the read, there is no kmer in the index that exactly matches the read. And since the read is not an exact match, it is sent out of storage for full read mapping. And finally, the filter moves on to the next read. So we can see that using the new data structures, GenStore-EM avoids random memory accesses. Also, the hardware cost is low because exact comparators are only using simple logic. Now here's a schematic of an SSD, including the design of GenStore-EM. Again, we have the three components: memory packages in orange, SSD controller in green, and internal DRAM in blue. Sorted reads and the sorted index of the reference genome are stored systematically in the memory packages, distributed over all channels and dies. The data structures are fetched sequentially by the SSD controller in batches and buffered in the internal DRAM. Comparison to filter exact matches is performed using a simple hardware comparator in the SSD controller. Reads that are not exact matches are sent out of storage for sequence alignment. Using this design, data fetching can be performed concurrently to filtering. And because the data structures are distributed over all channels, the full internal bandwidth of the SSD can be used. Now that we have seen GenStore-EM, let's also have a look at GenStore-NM. GenStore-NM filters reads in storage that do not align to any sequence in the reference genome. It performs the second step of read mapping, called chaining, and only sends reads for expensive sequence alignment that have a good enough chaining score. This filter is applicable to short or long reads with high error rate or genetic variation. Since this filter involves chaining, which uses dynamic programming, it's difficult to implement in storage, especially at low cost. When we look at the time and space complexity of chaining, we see that it scales quadratically with the number of seeds, which of course increases with longer reads. To tackle this challenge, the authors analyzed the relationship between the number of seeds in a read and the alignment probability. As you see in this figure, the more seeds there are in a read, the higher is the alignment probability. That gave the authors the idea to only perform chaining on reads with a low number of seeds. GenStore-NM sends reads with very good alignment probability out of storage for sequence alignment right away. On the other side, it disregards reads with too few seeds with no chance to align, and only performs chaining on the reads that have a number of seeds in between, thereby reducing the number of reads to do chaining on. Reads with a high chaining score leave storage for sequence alignment, while reads that have a low chaining score are filtered out. This way, if we recall the quadratic time and space complexity from the previous slide, the number of seeds n is at most the cutoff parameter, which is 64 here. This means that in-storage filtering of non-matching reads can be done at reasonable hardware cost. In order to evaluate the performance of GenStore, the authors compared state-of-the-art software and hardware read mappers for short and long reads, which are denoted with base, with integrations with GenStore inside the SSD, which is labeled GS in the next slides. They evaluated these systems for 3 different SSD configurations, a low end, a medium end, and a high end SSD that differed, for example, in bandwidth. So let's look at the results. We first look at the ones for GenStore-EM, and then I will also show those for GenStore-NM. To measure performance gain of GenStore-EM, a data set with short reads was used where 80% of reads were exact matches. Compared to both software and hardware read mappers, a measurable performance gain was achieved for all SSD configurations. But you can see that the performance gain was greatest for the low bandwidth SSD because a reduced number of reads are sent out of storage. The authors also evaluated the energy consumption by the system and found that compared to the baseline read mappers, GenStore reduces energy consumption. For GenStore-NM, the authors found even higher performance improvements, but using an extreme data set with over 99% non-matching reads. They also analyzed the performance gain for a data set with only 63% non-matching reads and found, as is to be expected, that performance gain of GenStore-NM increases with the percentage of non-matching reads in the data set. Here too, a considerable energy reduction could be measured. This brings me to the conclusion of the paper summary. The paper started from the problem that previous works that improve read mapping do not address the data movement overhead of moving data from storage to the rest of the system to perform read mapping on. The paper aimed at solving this problem by reducing the data movement overhead by designing an in-storage filter that filters reads that do not require sequence alignment. Challenges in this were differing workloads arising from varying read lengths and divergence from the reference genome, and limited hardware resources in the storage system. The authors came up with 2 filters, one for exactly matching reads and one for non-matching reads. And the results showed significant speed up and energy reduction compared to state-of-the-art software and hardware read mappers. So now let's move on to the paper analysis. The thing that I see as the main strength of this paper is that it really addresses the current limitations in data processing at the heart, the bottleneck of data movement. In this course, we have talked many times about the large fraction of execution time spent on data movement, not only in genome analysis. And solutions like the one in this paper show that moving pre-processing steps into memory or storage improve performance significantly and cost effectively. More specifically to genome analysis, this paper is the first to design an in-storage read mapping filter that successfully reduces the data movement overhead in addition to computational overhead. Also, in contrast to most previous works, GenStore is designed to filter a variety of different reads, short and long reads with low or high genetic variability. They achieve this using the 2 different GenStore subsystems. In the results section, we saw that GenStore significantly reduces energy consumption of read mapping and therefore facilitates genome analysis in portable devices where energy consumption is a key concern. And finally, the paper was a very good and clearly written read, even for a 4th semester student like me. All necessary background information, such as an overview of the read mapping process or the organization of a NAND flash-based SSD, was provided in the paper, which helped me a lot to understand the rest better. Now, after listing the many strengths of the paper, I will also discuss a few weaknesses that came to mind while reading it. Since I struggled most with this part of the analysis, I used the great list we saw in class to give me some ideas. As with all papers, this one could, of course, use more analysis, and clearly this impacts hardware, which is always bad. No, but honestly, even here, I had to struggle to find weaknesses, even though the list offers some low-hanging fruits. But since I was determined to find some flaws in the paper, I looked closely at the evaluation section. One thing I missed in the paper was if and how the two systems are meant to be used together. There are many data sets that would profit from applying both filters, but there's no description or much less an evaluation of such use cases. If both filtered are to be used together, for example, one after another on the same data set, how would they prevent the reads from the first filter to be sent out already for sequence alignment? So there's still some room to explore. Another thing I noticed was that the evaluation for GenStore-NM was done for a data set with 99.65% non-matching reads. This is obviously a very high percentage that can, for example, arise from using a reference genome from a completely different species. In this figure, the authors, which is also from the paper, the authors also analyzed the speed up GenStore-NM provides for data sets with lower percentages, here for 63% non-matching reads or 37% alignment rate. We can see that the speed up is much smaller, only 1.5x instead of more than 20x that we had before. And in my opinion, by choosing this extreme data set for the main analysis, the scale of the performance gain just appears a bit exaggerated. Another point is that they evaluate performance gain of GenStore-EM for a data set with 80% exact matches, close to what we have for human genomes. However, as read lengths might increase in the future, the probability for mismatches due to natural genetic variation increases and the number of exact matches decreases. So I did a quick calculation here. Humans have on average about one difference every 1,000 base pairs. And with a read length of 150 base pairs, this results in about 75% exactly matching reads. With a read length of 200 base pairs, this already goes down to 67%. And at 300 base pairs, we're at 55% of exactly matching reads. That means that less reads can be filtered out as exact matches. Also, genetic diversity is much higher in some other species, for example, viruses. If we assume a slightly higher diversity of one difference every 200 base pairs, the fraction of exact matches drops to 40%, and therefore performance of the method is lower for species with high diversity. That means that the performance gain of GenStore-EM varies highly with the application. But in fairness, I should say that variations in genome diversity is one of the reasons why they came up with two different systems. And as a last point, let's think back to how the authors addressed the many random accesses to the reference genome index. They increased the length of sequences in the index to be the same as the read length. Also, they sorted the reads and index and streamed them in for a comparison sequentially. However, even though the index representing the reference genome doesn't need to be present in its entirety in the internal DRAM, it still needs to be stored in the SSD. And by increasing the length of the index keys, the number of unique sequences of length k is also increased. To give some numbers, conventional read mappers use k equals 15, and the index structure for the human genome is 21 gigabytes. With k equals 150, which they used in the paper and which is also the current length of reads produced by very accurate sequencers, the index has a size of 126 gigabytes. They alleviate this by using not the original sequences, but a hashed fingerprint of both the reads and the index sequences, and manage to get the size back down to 32 gigabytes. But still, as lab tech companies such as Illumina strive to increase read lengths, increasing k accordingly always leads to an increase in storage needed for the index structure. But in my opinion, this is not a major weakness because storage capacity is cheap and becoming cheaper with time. This brings me to my last slide before the discussion. One thing I noticed throughout the paper was that even though data processing is in the computer science domain, it's also strongly connected to the current state of sequencing technology, which is governed by biological principles. So for me, it was surprising how much of the knowledge from my biology studies was relevant again in reviewing this paper. And now I also have an answer to why genome sequence analysis is limited by data processing. The problem, as in many other applications, is the discrepancy of advances in speeding up processors compared to memory. So genome analysis is just one more application where memory stalls determine the current performance limit. And now I'm looking at the time. We still have a few minutes left for discussion, and I want to conclude by thanking my mentors for their valuable help with my talk. So let's first start with any open questions you might have. Maybe anything from Zoom first until we have the microphone in the lecture hall set up, or?
Can you hear me?
Yes.
I was wondering, does the paper mention how the in storage filter is implemented? Do they modify the SSD controller or?
Yes, they add to the SSD controller.
Okay, thanks.
Then firstly, a follow-up question. Is any hardware necessary for the computations already in a standard SSD controller?
What do you mean, the computations?
Yeah, for the chaining. It's not as simple as comparisons, so that's...
No, it's not as simple as comparisons. So you refer to GenStore-NM, right?
Yes.
Yeah, there is hardware necessary, and it's necessary to kind of do the chaining, which is also done using software read mappers in storage. So it definitely needs hardware, and they try to limit the amount of hardware necessary in the area needed by limiting this parameter n to something they can control and say, okay, this can be done at reasonable hardware cost.
Okay, thanks.
Mm-hmm.
And then a second question about GenStore-EM in this case.
Mm-hmm.
We used those sorted arrays tables. Where and when is the sorting done? Because the paper mentions that the great benefit is that you don't need to move the data around, but sorting is more complex. So can you do sorting in SSD, or how is that done?
Yeah, thanks for that question. No, it says that it's supposed to be done before. So it gets sorted and then stored in this data structure in the SSD, but it's not part of the GenStore algorithm. It's a prerequisition that it be sorted the way. I can go back to the slide. 1 second.
So the reference genome would be already sorted?
Yes, both would also already have to be sorted and stored in this method in the SSD. So this means, of course, it has the, if you want to call it, weakness or disadvantage that you have to do it before, but no, it's not part of the GenStore process or algorithm or architecture to do this. So they don't talk about implementing sorting also in the SSD.
Okay, thanks.
Mm-hmm.
Thanks, Jennifer, for your response. I would like to also add a little bit more detail about what you already described. So yes, the sorting is a pre-processing step, but what we envision is that since already when the genomic data, the reads are being generated, they're being generated in different batches and they go through a pipeline. So they go through sequencing, then they go through base calling so that the sequencing output would be turned into DNA alphabet, the ACGT. So we envision that sorting would be another step in the same pipeline so that different batches that come, they will go through these steps. And the throughput for sorting, we show that is much higher than the throughput for base calling and sequencing. So overall, this pre-processing step would not change the throughput of the pipeline which is used for genomic read data generation. So yeah, I just wanted to add that. And we have more details also about that in the paper, which you can refer exactly to the throughput of these units and how this sorting, in a sense, for a batch can be masked by other parts that are already being done.
Yeah, thank you, Nika. This is a good comment that the sorting process is not limiting in the pipeline. Yeah, I forgot to mention that. Thank you. Any other questions?
Hi, so thank you for the very interesting review. Going back to the size of the index, it's something that also has surprised me, and you pointed it out, that the decision to use the K-mer length of 150, I'm actually quite surprised that this index size hasn't increased more dramatically, because if I'm not mistaken, the size should be basically exponentially depending on the length.
No, I thought that at first too, but it's about unique sequences of length k. So of course it is a bit longer, but it's not exponentially. It just depends on what kind of sequences there are.
Right. So they don't have all the possible sequences, I guess.
No. Maybe I can show this, for example, here. You have it ends with A, it ends with C, it ends with T, but not with G. It's really the actual K-mers you have for which you have locations, and that actually exists. So it's hard to predict the number of or the size of the index structure just by increasing the length. But I think as a rule of thumb, you could say that it gets longer because there are just more unique sequences of that length.
Yeah, of course. And second of all, with the growing size of the K-mers, isn't there like a high probability that the reads are not perfectly aligned with the K-mers, therefore essentially causing a lot more mismatches?
Yeah, this is what I tried to display here. I just calculated, you have the error rate of 0.1%, and I also included error rate of 0.1% from the sequencer. So I just, how I came to these numbers is just by calculating 0.998 to the power of 150 to the power of 200 and so on. But this is exactly what you see is just by natural genetic diversity. The longer the read, the more likely it is that you just have a difference, a natural difference to the reference genome.
Yes, also, but what I meant was the alignments of the the reads with respect to the read sequences. There is a higher probability that they can be misaligned, or is that not an issue?
What do you mean, the read to the read alignment? I don't understand.
So you've got your K-mers and you've got your reads. Now you're trying to match them now. The way I understand it, there's a huge probability, the longer the read is, that your read is not perfectly aligned to the K-mers.
Yeah, maybe I'm not sure if I understand completely, but the K-mers are the reference genome, are the unique index, unique sequences of length k in the reference genome, and they just store it as an index. Maybe I can go back to this one here. So you have the reference genome here, and you have, for example, GCC at location 1, 2, 3, 4, 5, 6, 7. So it starts here, and so on. So the K-mers are the reference genome.
Yes, but for a smaller K-mer, there's like less of a chance that your read is going to somehow be misaligned. Maybe it's just the implementation detail. It's just interesting to did they do any analysis, or why they chose to pick the entire length 150 for the K-mers, or is it just because the hardware is simpler, or
yeah, it's because of these random accesses, as I tried to explain. If you choose the entire read as the thing you look up, you just have to do it once and have to look up the entire read instead of choosing subsequences which appear to be random, and then you have to do random accesses to the index.
I see.
No.
I see.
Hmm.
Okay, thank you.
Yeah.
It's a very good question.
I would. Oh, sorry, Mohammed.
Oh yeah,
Please go ahead.
Want to comment? Go ahead.
Oh, okay, thanks, Mohammed. Thanks, Jennifer, for the responses. Again, some details to add. So regarding the first question about the sizes, yes, the sizes increase. As Jennifer also mentioned, exactly exponentially, but based on what is really present in the reference genome. But also we further reduce the size of the index by replacing the read size k-mers with the cryptographic hash values as their fingerprints. So we use some fingerprints instead of storing the whole read as in each entry of the index. So that significantly reduces the size of the index. And another point regarding misalignment with the larger sizes. So no, that does not happen because we extract every single read size k-mers from the reference genome. So if the read would match to any place in the reference genome, it would also match to a k-mer in our new index.
But he's also asking, what if it doesn't match to a read long k-mer? What will happen? Will you miss a line or miss the read for good? I think because we maintain the two steps still, you do filtering in SSD, and if it doesn't exactly match, you still continue with the read mapping process. So if it doesn't match, you extract the short k-mers as usual, then you try to align it to somewhere else. But that happens in the CPU as usual read mapping. But the great benefits will happen when you filter a large portion of the reads inside the SSD as if they exactly match. And again, this exact matching filter only for short reads, not for long reads.
Yeah, and it really profits from being able to use these the simple logic for exact matches and having no mismatches, not even one or two, to be able to kind of allow. At the moment, it really is for exact matches. So as it gets longer, it might have a smaller percentage of reads. Any other questions?
Yeah, I had another question.
Mm-hmm.
I've noticed that for the EM model, we keep in the memory the snippets that have an exact matching, while for the NM model, we send the sequence we identify out of the memory. Why is there a difference? I mean, why do we keep the exact matches in memory, while in the other model, the one that we have to further analyze are sent out of the memory? Does the article explain that?
Yeah, so the system for exact I mean, it's a filter, right? So you want to decide what stays in storage, and is where are you able to decide that it does not need sequence alignment. And exact matches don't need sequence alignment because you know the locations already from the index for which you match. So you filter those out, meaning they don't go out of storage. But for GenStore-NM, you want to filter those out that do not align to any sequence. And those are the ones with the either, let me go back to the slide. I understand I think I understand your question, that it's kind of confusing. So here for GenStore-NM, they send those out that already have very many seeds, so sequences aligning to them from the beginning, because they have very high probability of having of needing sequence alignment, of performing good in sequence alignment. So they don't do chaining on those, just send them out right away, and only do chaining on the ones where they're not sure if they're good enough to qualify for sequence alignment. And they filter those out with a bad chaining score, which are the ones that would not align to any sequence. So you have both extremes. The best ones are the exact matches, and the worst ones or the bad ones are the ones that do not align to anything. And it only makes sense to do sequence alignment, which is so expensive for the middle part. Does that answer the question?
Yeah, okay, thanks.
Mm.
I have a question for, very generally, what are the difference between doing that in an SSD and doing that in memory? Like with processing in memory, could that be, is that too expensive or?
Yeah, I mean, you have the data, the genomic data, which is very large, too large to just load it all into DRAM. You have it in the SSD. And as I said, it's too big to have it all in the normal DRAM. So you would have to move the data from the SSD to DRAM to do the filtering in there. But of course, it makes sense to, or would make sense to also filter in DRAM, but then you would still have the data movement overhead.
Okay, but I mean, if you're creating specialized hardware, this is probably pretty expensive already, right? And
in terms of what expensive?
to
I mean,
produce?
it's not specialized hardware. It just adds to an existing SSD. So you have the normal SSD. It's actually still able to work as a normal SSD, and you add to the hardware in the SSD. And in terms of how much space or area this needs, this is not very expensive. Even if it's envisioned to have both systems, GenStore-EM and NM in an SSD, but still it doesn't need a lot of additional area.
Oh, okay.
Mm.
Thanks.
Nika, you correct me if I say something that.
No, that makes sense. Yeah,
Okay.
that is correct. And also, something to think about is that, so imagine you want to filter some data from some process. Then you have the option of filtering it inside the SSD or inside your memory or inside an accelerator, I don't know, outside memory. So if we can filter the data as early as possible for the data that is already located in the storage, then we kind of like get rid of the data movement overhead right up from beginning. So we don't need to move the data from storage, which it is already residing in there, to memory to do in-memory processing or like PIM. That's why we thought that, okay, filtering it inside the storage would provide the most benefits, also because of the I/O overhead that we showed. And then this design, GenStore, can be also integrated with a PIM solution that is accelerating the next stages of the read mapping, because concurrently inside the storage, they can filter things, while a PIM design or another accelerator can accelerate other steps of the read mapping. I hope that answers your question also.
Yes, thanks. So it's also more energy efficient.
Yes.
Okay.
Yeah.
Thanks.
Any more questions? Yeah.
Yes, on one of the first weakness I think you mentioned about the paper.
Yeah, let me go back to the slide. Here
Was
about,
said,
yeah.
Yes, there's no description or evaluation of using both filters together. And my question would be why? Because like my idea is that maybe, and you have the background knowledge, you know in advance what kind of data you have, so you know that only one filter will achieve the great performance increase, and then using the other one also would just mean computation overhead. And if you can't answer, then maybe to the authors why there's no, why it's not proposed.
Yeah, I discussed this a bit with Nika because I had the same question, and she explained that they envision it to have both filters in the SSD, and the programmer knows the data set and can choose which one they want to use. And in some data sets, only one of the two filters makes sense to use, and then you can use this. Or there are some data sets where you can use them together. And for this kind of the evaluation is missing, yeah, or at least I had some questions. Okay, how exactly would you use this? I mean, they don't, as I said, one would start and send out the sequences already, but then they're out, right? And then only the other filter comes next. So this is what's missing. But at the moment, they plan it to have the programmer decide which filter they want to use.
Okay, thanks.
Yes, and on top of that, they can run, as Jennifer also correctly mentioned, they can run sequentially back to back. And since the execution time of each of these filters is very small, then together they can run meanwhile in this SSD, while the unfiltered reads can go to the read mapping in wherever the read mapper is located, and then concurrently the read mapper can work while these two sequentially can execute in the SSD. So most probably the execution time of both of them would be hidden or like fully overlapped by the read mapper. But of course, the exact timing of it, it is a good result that would that a paper would benefit from it, or like something to quantitatively show. But that is the way we envision it to work.
Okay, then maybe one follow-up. Can you, using both filters together, can you prevent one filter from sending the data directly to a host, but instead to the other filter?
Yes, yes. So
okay, then it makes sense. Thanks.
Yeah. But definitely, it would have been good to include it in the paper. So I agree with Jennifer including it in the weaknesses.
So I have two more questions. So first of all, since you have so many, or you have multiple data banks in the memory cells, are they using any kind of parallel accessing? You mentioned that they can exploit the full bandwidth, the internal bandwidth of the SSD, but are they maybe like reading from multiple banks at the same time, or how is this performed? They discuss this in the paper?
Yes, let me go back to the graphic here. Yes, as I understood it, this is the idea that you have or maybe I can show it, the different one that shows that the channels here, as I understand it, the channels are really separated with these different flash controllers. So it is possible, and that's also the idea, to have it in parallel, which is why it's distributed over the different channels and dies.
So it seems like since the computation is so simple, like it would be quite simple to just run all of them in parallel. And second of all, have they tried to sell this to any other applications, such as, I don't know, natural language processing or some kind of?
Oh, I don't know, but my first thought is this was in March, so I'm not sure if it's if this is still already going on, that they kind of improve this further. But I'm not sure. Maybe Nika or Mohammed can say more about this.
So yeah, I mean, we also somewhere in the, like maybe in the conclusion or some ending parts, we briefly mentioned that we hope that the key ideas or optimizations would be also used in different genomic applications or different domains. So yeah, so for different applications that work on like pattern matching or other IO-intensive applications, because some of these primitives, such as the sorting or the type of data mapping that we used or other optimizations, we believe that can be used in other domains as well. And one of the immediate applications that we want to look into would be using or like expanding this to metagenomics. So that is something we are interested in.
Yeah, thanks. Thank you. Any other questions? I think that's it. We're already all tired.
Yeah, I had some discussion topics prepared, but I'm looking at the time and I think it's time to call it a night, if that's all right with the course instructors.
Yeah, I think they might have already left.
Okay, all right.
No,
thanks.
very interesting.
You
Thank you
can
very
keep
much.
going if you like.
Okay, in that case, thank you everyone for listening and have a good evening.
Thank you so much, Jennifer. Thanks, Gianluca, for the two interesting talks.